That's right. So hi, everyone. So yeah, our tour of the subcortex ends right now, but we're not yet moving to the neocortex. So first we'll stop at the archicortex or hippocampus. So this has been our view of the hippocampus for a very long time. So this is just a Golgi stain from a rodent that I've oriented. So I flipped it so that it resembles the human orientation. And basically, we have these huge pyramidal neurons that are organized into what we call subfields based on their morphology. So for a long time, the idea was that in the human brain, we would take a cross section like this one, and we would just see the same structure repeated across the whole anterior, posterior extent of the hippocampus. And so people have now tried to infer the location of these subfields in MRI. And typically, you would do that and get something like this. So here, this at the bottom right here is just a coronal section from an MRI image. And they've tried to use a volumetric registration to infer where each of the subfields might be, although we don't have the histological features to actually inform that decision. So it's basically using a registration-based approach. And that gives you kind of a rough picture of the hippocampus, as you can see on this 3D model on the left here. But I want to show a little bit more detail of the hippocampus. So first, let's jump into big brain. So here is just a typical coronal section. Here, how do I use the pointer? Yeah. Okay, coronal section of the hippocampus in big brain. And we see this sort of classic C shape. Maybe quick note here, the subiculum could be extended further, but we just cut it off here for pragmatic reasons that have to do with translating to MRI. So this is, again, this agrees with our idea from histology. But when we start to take slices from different parts of the hippocampus, so here's a slice through the anterior or the head of the hippocampus, we can see that it doesn't look like a classic sort of C shape anymore. Instead, we see a pretty complex arrangement of these different subfields. And same story if we take a slice through the posterior or the tail of the hippocampus. So this makes the story quite a bit more complicated. And I'll also just show one sagittal section through the hippocampus as well, where again, it's not totally clear how these subfields relate to each other spatially. However, in big brain, because of this 3D and digital nature of the data, we can actually dynamically slice in all sorts of different directions here. And so I'd like to highlight a fairly simple principle for understanding the morphology of the hippocampus, which is basically that across the different subfields, we always go from dentate gyrus down through the CA fields to the subicular complex. So just looking at a slice through the anterior hippocampus, which is shown up here, so a slice through the uncus taken at the right angle, you can see the same ordering of subfields. So dentate gyrus to the CA fields to subiculum to entorhinal cortex. That's in the very anterior uncus of the hippocampus. You can also slice at different points along the hippocampus. So here, you should see the same ordering of subfields again. And basically, if you sort of take into account the gross curvature of the hippocampus, we'll see that the arrangement of subfields is always consistent. It always follows the same topology. And so this is one simple organizing principle. Part of the issue, though, is, so here's just a few other hippocampal surfaces sampled from MRI. And you can see this is actually a little bit more complicated because different people have very different folding or morphology within their hippocampus. So overall, we have a bit of a complicated task ahead of us if we want to understand the structure in MRI. And the method that I'd like to propose for handling this is very similar to what we do in the neocortex, which is basically, even though people have different gyral and sulcal patterning, we can inflate that to a sphere and then register individuals on a sphere in a topologically constrained manner. And so what I'm proposing to do is take our hippocampus and unfold it to what I'm calling here this unfolded space. So basically along its proximal to distal and anterior posterior axes. So I'll flesh that out in a few examples. And so, yeah, basically the proposal is, even though we have variability in the shape of the overall hippocampus and its folding, we hopefully can map always to a consistent unfolded space analogous to the inflated sphere. So now I'd like to just walk through a few empirical examples where we look at this in more detail. So first, just within big brain itself. So Probably some of you guys are familiar with this already, but basically we can extract more detailed features within our segmentation of the structure. So we can look at things like the thickness, the curvature, the surface textures, or the extent of gyrification within any piece of cortex, but in this case, it will be the hippocampus. And so here I've just mapped all these features to the unfolded hippocampus. And so you can see across the different points of the hippocampus, we see fairly consistent differences. For example, the thickness being much higher in the subiculum than the other subfields. We then additionally looked at these laminar profiles, so going basically from the inner to the outer surface of cortex, in this case hippocampus, and we can summarize that profile with a few moments, and also then take the absolute derivative of that and summarize that. And so altogether, this gives us a huge set of features that describe the laminar organization of the hippocampus. And so what I want you to note from all of these features is that I've overlaid manually defined subfields in these white lines, and these features tend to closely match our manual definition of subfields. So what I did then next is basically take all of these features and use an unsupervised clustering method, so PCA followed by k-means clustering. And when we do that, we get these clusters that closely resemble our manually defined idea of what hippocampal subfields should look like in a totally unsupervised way. I'll also just highlight a sort of similar study by Piccola et al., where they looked not only at the hippocampus, but then also extended this over the neighboring neocortex or isocortex, if you like. And here again, they looked at these laminar profiles and computed their similarity to one another. And so again, within the hippocampus itself, you can see differences along where the different subfields would be. And then these differences basically continuously spread into the surrounding isocortex. And so they refer to this as the confluence of iso and allocortex, that is neocortex or hippocampus. So yeah, hopefully that gives you this idea that from what I'm calling proximal to distal, so basically along you could also think of it as medial lateral aspect of the hippocampus, we see this structural difference that basically we usually refer to that in histology as being the subfields. I'll talk a little bit about some MRI-based work. So first of all, how do we go from big brain to MRI? So this is sort of how the unfolding works in big brain. We start out with just a tissue classification up here, and then we impose these geodesic coordinates that then make up a 2D indexing system for the unfolded space. So in MRI, we take an image like this, we crop around the hippocampus. I've been implementing a convolutional neural network to then get basically these same tissue classes, but they are now subject specific. So basically we have this shape of hippocampus, which we then impose the same coordinates on, and in that way we translate it to the same unfolded space. So this is just looking at a gross set of results from, I think it's about 100 subjects from the HCP data set. So if we just look on average, the hippocampus, once we average it over many people, is quite smooth, just because no two people show exactly the same folding pattern. But some of it is conserved, so you see this overall sort of shape. And then you can map out a whole set of features, so I won't go over all of them, but for example, you can just look at intracortical myelin and see that it differs primarily across the different subfields, so along where we see those subfield boundaries again. And I'll mention really quickly some work under review by a friend of mine, Brad Carrot, who is mapping this out in diffusion-weighted imaging and basically looking at across the anterior to posterior versus the sort of proximal distal and also the inner to outer, so that is the thickness of that tissue, whether that aligns to the different diffusion directions. And again, what we're seeing is these structural differences that closely align with the different subfields. So basically moving from proximal to distal. So again, in MRI, we see the same sort of structural differentiation of a simple proximal to distal axis, again, separating the subfields. And also come talk to me if you'd like to try this unfolding yourself, which is available in this PIDS app. And now a little bit of resting state fMRI. So the conclusion here is that it's actually a very different story. We see differentiation across the anterior posterior axis of the hippocampus. So basically the way that we do this is first we map some time series data onto the folded and unfolded hippocampus, which I'm showing here. And then we look at the functional connectivity between all of these hippocampal points or vertices to corresponding neocortical points. And we then just zoom in on the differences between, well, the connectivity between hippocampus and neocortex. And we then do some dimensionality reduction, in this case, using a tool called BrainSpace. And so we see here the primary axis of differentiation fits almost perfectly this anterior posterior geodesic axis in the unfolded hippocampus. And it's a sort of more curved, complicated configuration in its native space. But in unfolded space, it's very simply described as anterior to posterior. And then we can also then just look at where those projections are in the corresponding neocortical areas. So for example, anterior hippocampus, which is shown in the very bright colors here, has a corresponding functional connectivity to inferior frontal cortex, and posterior hippocampus more connected to visual cortex. I'll show one other quick study. So these are some gradients describing neocortical connectivity. So this is known as the sensory transmodal gradient. And in Casey Piccola's work, they also looked at connectivity of the anterior versus posterior hippocampus and extended neighboring cortex. And they saw that basically they were almost perfectly aligned with one another, such that the anterior hippocampus was more connected to transmodal regions, and posterior hippocampus and surrounding cortex were more connected to sensory regions. Quick caveat though, that this work was done in volumetric space. And so at that point, we hadn't yet done this sort of topological mapping between different subjects. So a bit of a caveat there. So hopefully that should show you that even though structurally we see most of this proximal distal differentiation, functionally we see almost perfectly parallel or orthogonal anterior posterior axis of differentiation. So altogether, what I'm sort of proposing here is a very simple model of the hippocampus. Of course, there's nuance and details that's not captured here, but the overall conclusion is very simply that from this proximal distal axis, we see structural differentiation, whereas across the anterior posterior geodesic axis, we see structural differences. There's been a lot of other sort of work on this in the literature that I think has maybe been slightly confused by this inability to look at slices in different planes or consider the overall folded or curved shape of the hippocampus. And so what I want to show you guys is just a very simple model of the hippocampus that hopefully you agree fits both big brain data and fits with MRI-based observations as well. So with that, I'd like to thank everybody who contributed, and I'm happy to take any questions.